Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension Cooperative Extension, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Russ Groves from the Department of Entomology. He was born and raised in Oskaloosa, Iowa, where he went to high school. He went to Ames at Iowa State University for his undergraduate, went to Fayetteville at the University of Arkansas for his master's, and then went to North Carolina State University in Raleigh for his PhD. He did a postdoc at Cornell, and then spent several years working for the U.S. Department of Agriculture, otherwise known as the Agriculture Research Service, in Kearney, California, working on sharpshooter. And then he came to Madison, where he's both in the UW-Madison Department of Entomology and a state specialist with UW Extension Cooperative Extension. Tonight, he gets to talk with us about pest management in Wisconsin's specialty crops. And if you want to know what a specialty crop is, go beyond corn and soybeans and alfalfa, and that gets you into things like potatoes and winter wheat and barley and oats and corn, sweet corn, cranberries, peas for packing, splendid things like that that make life a lot more pleasant. They may not be huge in acreage, but they're really important to our culture and to our tables. Please join me in welcoming Russ Groves to Wednesday Night at the Lab.
Well, Tom, thanks a bunch. And it really is it's a pleasure to be here, and I appreciate you all coming out to listen to this presentation. As I told Tom when we just visited, what I'm going to talk to you about today is a bit of a work in progress. I wouldn't have said 3 or 4 years ago that I would have been moving down this trajectory or this research line, but I think it's an important topic area to talk about. And I think for our Wisconsin specialty crop producers, it's something that I think they understand too is an important message to convey. Before I go too much further, I really want to focus on the people that actually do the work in my lab. So as Tom had indicated, I'm an appointment between UW Extension and CALS. As an integrated position, my title as state specialist is vegetable entomologist. So I have responsibility for vegetable pest management here at the UW. My research area of emphasis primarily has been vector-borne disease. So we focus largely on insects that transmit pathogens, viruses, bacteria, phytoplasmas. But as part of my work as the vegetable extension specialist and learning how to control insects, we sort of went this direction. And one of the people that I want to highlight tonight is Ben Bradford, which for you is the gentleman wearing the goofy hat in the upper left. Some of Ben's work, and then a person who is currently a postdoc at NC State University, Anders Huseth, who's actually grew up in Rio, Wisconsin, and received his PhD here at the UW, another individual whose research we'll highlight. But before we start, and I should say, really what I want to do is I kind of want to talk you through a little bit of Wisconsin specialty crop agriculture, why it's important. I want to kind of take you or segue into a little bit of the work that I would do on a day-to-day basis, and that is learn how to control an insect pest that is causes us issue, if you will, and then kind of lead you along the path that I was led as far as learning about the consequences of the management of that pest. But at least out of a text, I think it's important. This gentleman, Dr. Marshall Johnson, is an emeritus faculty at the University of California, Riverside. Marshall's actually a personal friend of mine, and he's a colleague and also a collaborator of mine who published in a book titled Challenges Within Entomology, a Celebration of the Past 100 Years and a Look to the Next Century. And Marshall writes, military metaphors are often used to describe our attempts to suppress or manage arthropod populations. Titles of both lay and scientific articles relating to arthropod suppression often include military terms such as battle, campaign, defense, enemy, and war. L.O. Howard was probably one of the best-known entomologists to commonly use these military metaphors in his writing. And recently in a book titled Nature Wars, People versus Pests, Winston stated that pest management has become a modern war against nature. But it is time to reconsider the terms of engagement. And I think that's a little bit relevant to some of the things that we'll talk about here today. And I'll try to conclude politely and respectfully with a bit of a perspective that I think is also somewhat near and dear to Tom's heart. So this is kind of Wisconsin specialty crop agriculture. I'd like to say that Tom underestimated Wisconsin specialty crop agriculture. Yes, it is not corn, cotton, rice, wheat, and beans. And by beans, I mean soybeans. But it is this reasonably significant outlay. In fact, Wisconsin is the second largest vegetable processing producer in the country. We are the leading producer of crops including green beans, sweet corn, cranberries. We are third nationally behind, in potatoes rather, behind a few states which will remain nameless because I just don't think it's worth mentioning their names. Interestingly enough, we are the world's leading producer of ginseng in Marathon County. Probably many of you knew this. And not mentioned there, but we are the world's, or rather the national leader in the production of sauerkraut. And that comes out of Bear Creek or Shiocton, the Shiocton area. In fact, Ryan Downs, I'll depart just a moment, if you'll allow me. Ryan Downs, the proprietor, would say that every time he sees a hearse go by, he says, "There goes another customer," is how he would describe that. He would then turn to me and say, "So Russ, where do you think the majority of sauerkraut is consumed in the country?" And I would then erroneously say, "Well, Wisconsin, of course." And he said, "You didn't hear me." He said, "Every time I see a hearse go by, there goes another customer." I said, "Oh, well, that's Florida and Arizona." And he said, "Precisely." So, but that is Wisconsin specialty crop agriculture. This is what the USDA NASS describes as specialty crop agriculture. And I don't think it would be any surprise to you that you would know California, of course, is a fresh and processed center. It is number one in vegetable production. But we have historically had significant production in the winter garden area of Texas, South Texas, Florida, the Delmarva area. Lots of potatoes growing in those unnamed states in the Pacific Northwest. But then you can see there's lots of blue dot-ish in the upper Midwest. In fact, recently, the current executive branch of the government has identified the upper Midwest as an area of national security for food production. And some of the reasons for this, there really is a sort of a changing economic ground. There are retailers that can shape or reshape production. And even through public policy with respect to even sustainability standards, retailers that are jumping in front of each other to, I would hazard to say, green themselves up a bit, are making claims about what they can do to a buyer, or what they can offer you as a buyer. And we then respond to that. You've probably heard in the news of late, unfortunately, even access to water is becoming a huge issue with respect to food and food production and where it will occur. And even fuel prices. Fuel prices in 2008, when diesel had reached about $5.50, was really having a huge impact on our ability to move fresh cut greens and lettuces from the West Coast to the East Coast, and moving these at a loss. So these are just some of these issues that are affecting production. Some of the other issues that actually affect Wisconsin pest management, and specifically my recommendations, can even be the major retailers. You probably understand that about 75% of us are fed by about 4 principal retailers, right? And number one, do you know who number one is?
Walmart.
Yes. And since we're being taped, I won't make any comment further. But, and when Walmart speaks, growers will respond. And so if a major retailer says that their now competitive advantage is no detectable residues, then the days of our ability to use an organochlorine or a carbamate or an organophosphate or an older insecticide is generally done, is over. So in many ways, that's not a bad thing. And we have to learn how to use reduced risk insecticides or even biopesticides. We export a lot of commodity. And even a lot of our export buyers are expecting and demanding safe products. So again, our choice as far as a recommendation is guided by the endpoint and the endpoint market. And as I mentioned before, water quantity and water quality is now becoming a very significant issue. It's becoming a very significant issue even in Wisconsin. Interestingly enough, a lot of the targeted water delivery systems through drip irrigation and so forth afford us the opportunity now to even deliver novel pest management through these types of devices, which is actually a very good thing. So this is really IPM. IPM is the integration, or the approach that we take to managing a pest is something we would call integrated pest management. And it is the integration of some or all of these approaches to achieve a level of pest prevalence at or below an acceptable level, right? And the components of an IPM program are first and foremost that someone is out there actually looking at the crop and seeing that there's a pest present, identifying that pest, making a decision about what we should do, then intervening. And thankfully, the majority of pest managers in this country don't do the last 3, and then I can have a job. But in fact, they should follow up, right? They should learn whether or not what course of action they took was appropriate and then educate oneself. And Vern Stern, in 1959, an entomologist, indicated that IPM is really just an approach that applies ecological principles in utilizing biological and chemical control against insect pests. Now, Vern was probably the instigator or initiator, if you will, of IPM, but it has become more contemporary, and now the Council on Environmental Quality has gone so far as to say it is an economically efficient and environmentally preferable approach to pest management that is regarded as a decision aid. And by that I mean it's our choice to decide along that pinwheel what components we put together to choose that are sustainable, environmentally safe, and socially sound, right? So that is really sort of what IPM is. And I kind of want you to think about that, and we'll come back to that later in the presentation. So now let me take you sort of through a day in the life of a vegetable entomologist. And when we manage a pest in a crop like potato, we don't focus on every pest that's there. We typically focus on the key pests. And the key pests in potato are the Colorado potato beetle, a potato leafhopper, and colonizing aphids. And we fashion our program around managing the key pests. And we're going to focus on one of those today, and that is the Colorado potato beetle. One of the first things that somebody has to do, to some extent, as in this dialogue, with, again, our war with insects, is know your enemy, right? So we have to know the life history and the biology of this pest. So right now, Colorado potato beetles are digging deep. They're burrowing into the soil right now. They're burrowing into areas outside the field under mulch, under tree lines, and going anywhere from maybe 20 to as much as 30 inches deep. Now, they can average 6 to 12 inches deep, but there are certainly insects that are getting that deep. The reason being they're wanting to get ahead of the frost line, right, that develops. If you try to overwinter in the middle of that field, you probably won't make it past about the middle of January. But so most of the insects overwinter outside the field as adults. They then emerge out of the ground when the frost disappears and they feel the warm rains moving through the soil profile, and out they come as adults. And those adults really don't have that much of an appetite. They've been in the ground for all winter, and they have just a little bit of energy, and they have just enough energy to find either a girlfriend or a boyfriend, and then they lay the majority of their eggs, right? And then once they've done that, those adults perish. Those eggs hatch into small larvae, and they feed for a period of about 20 to 25 days, going through 4 larval development stages, which we call instars. And what you don't want them to get to is that latter 3rd or 4th instar stage, because that latter 3rd or 4th instar stage is very challenging to control. And then after they've fed a bit longer, like all holometabolous insects that undergo 4 metamorphic stages, they pupate in the ground, and then they'll graciously reemerge as adults to continue that cycle. Now, unlike their parents, who weren't that hungry, these adults are hungry as the dickens, and they will feed quite voraciously. And they'll feed through July, and then they'll finally continue feeding until they see enough environmental stimulus, declining daylight and declining temperatures, and down in the ground they go again, right? So we've learned about the seasonal ecology of this insect, right? They overwinter. In the spring they emerge, they walk to the crop. They have, it depends on the year, a couple of generations per year. And the larvae have historically been the principal stage that we have used insecticides to target. But more recently, and since 1994, which was the registration of the first neonicotinoid insecticide, which has very successfully killed almost all stages with the exception of an egg stage, which is a non-feeding stage, this set of tools have been very successful. So I might ask the audience, has anyone heard of a neonicotinoid insecticide? Right. Okay. And keep your hands up if you've heard of it in a good light. That's what I thought. And so this is another way to look at this. The above panel is the phenology of the adults. The middle panel is the phenology of the eggs that they deposit. The lower panel is the phenology of the small and then later large larvae and then pupae in the ground. And really, the value of the neonicotinoid insecticides were such that they could place them in the ground at planting. They would become water-soluble, systemically mobile, and move throughout the plant and provide almost season-long protection. In fact, they were registered in 1995. And on this illustration, you can see, here's 1995. Well, before 1995, this insect, which on the poster children for resistant insects, it sits next to diamondback moth and Colorado potato beetle are probably the 2 insects in the world that have the highest potential for developing insecticide resistance. You can see that before the registration of the neonics in 95, growers might have been putting on as many as 10 to 12 insecticide sprays in a season with an environmental impact quotient score of 4.7. More is bad. When the neonics were registered, one single application in the ground, problem solved. Our EIQ score plummeted to 0.3. So now I was hired in 2006, and I could argue that all hell broke loose. because here we were emerging back to using more pesticides. And the reality is that we are having resistance to the neonicotinoid insecticides. But because of their broad spectrum nature, they're still a very valuable tool to have in place for control of the other key pests, right? And we're actually now able to fashion pest management programs more contemporarily that use these foliar reduced risk insecticides that can drop our EIQ score even further. But I will say that a neonic is still part of that program for the reason I just described. So here's the group. This is the Insecticide Resistance Action Committee's classification. They are a group for a neonicotinoid. And there are, well, you can see even nicotine is in that group loosely. But the neonics principally comprise about three principal uses, or three principal registrations. Clothianidin, imidacloprid, and thiomethoxam constitute the majority of national uses, right? At the time they were registered, they were unquestionably considered reduced risk because they were organochlorine, organophosphate, and carbamate alternatives. And there is no question, from acute and chronic DEEM analyses, which are the manner in which EPA evaluates the impacts of these were by far worlds better. They are effective and long-lasting systemic insecticides, right? And in fact, right now, this class constitutes as much as 25% of the national market of pesticides. Pesticides. Not insecticides, pesticides, right? A very used class. So what I mean by when we talk about systemic insecticides, my point here is that if you put this insecticide in the ground, it will move acropedally. It will move up the vasculature. And the illustration on the right is just a label that illustrates where the compound is generally distributed. And you can see it's not terribly evenly distributed, but it is generally distributed through the plant, right? So that's what I mean by an ease of use, is that these compounds, again, we went from 10 to a dozen sprays down to one, right? One at-plant spray, 1994 until now. So we've had 20 years of use, right? These in-furrow management approaches have now been adopted on over 85% of the acres in Wisconsin. That's a very conservative estimate. And I would say that, unfortunately, there are really no products on the horizon that have the same spectrum of activity or ease of use. There's not that many products that are systemically mobile and have that kind of residual activity for an industry. These growers were seeing 85 to 90, in some cases 95 or 100 days of residual activity out of one application. But because resistance is prevalent, we're now having to switch away from their use for targeting this insect. But they are still in the system, remember, because they're successful against the other key pests. And I mentioned that, part of the reason is just because of the decorated history of the Colorado potato beetle. This is just an illustration showing you what the Long Island, New York populations of Colorado potato beetle are capable of. When the initial carbamate carbaryl, which you might know as 7, was initially registered in 1957, you can see it lasted quite a long time in New York. It's a joke. Lasted a year. You can see actually glutathione, which is azinphos-methyl, and organophosphate, woohoo, 5 years. But other members that were registered just in the few years that follow had no chance, right? And remember the concept of sort of island biogeography. If you are isolating all of these genetics on one island, have no opportunity for having susceptibles come back into that population, this is what you get, right? And in fact, you can see now down here at the very end, the neonics, imidacloprid and thiomethoxam, lasted quite a long time, in fact, on on Long Island against this insect pest. This is just an illustration, if you go to this URL, of what the USGS has as far as proposed estimates for use for one of those neonicotinoids, and that's clothianidin. The leftmost panel is 2003, the rightmost panel is 2009. In a 6-year time frame, you can see that the extent of use of these insecticides, I might go so far as to say, has skyrocketed. Principally, these are seed treatments, and I apologize if you can't see. The lower panels show you by year and crop ID what they're principally used on. And the yellow bar there is corn, and that is a corn seed treatment. This is thiomethoxam that has a bit more of a broad registration, and it is labeled on corn, soybeans, and the red is cotton. But potatoes and other veggies, I would say, pale in comparison as far as the extent of use of these neonicotinoids, and you can see they have a fairly extensive use nationally. So, remember, these are water-soluble and systemically mobile insecticides. And in Wisconsin, we're now having issues with respect to groundwater quantity, right? Groundwater quantity. These conversations are going on even in the Central Sands production area about reductions in our water resource. And there are many users, there's no question. And we do have to ask the question about how sustainable this resource is and really the focus that we have here. And they will probably or likely require complex solutions because water is a critical component of the history and the future of Wisconsin. There is no question. And this is, if you were to Google water in central Wisconsin, some of the first hits that you might find. And this is the Little Plover River in central Wisconsin. There's no question this is unfortunate. The industry is making attempts, make no mistake, to remedy this. They recognize that they are in part liable for this. And they are making those gestures. But it was in about the third or fourth year of my time here at UW when we began the process of working more closely with the Department of Agriculture, DATCAP more specifically, the Wisconsin Department of Agriculture, Trade, and Consumer Protection. And with their laboratory, the analytical lab, and I have to thank them significantly because they were initially the first set of individuals that helped us to start to analyze these data. We started to see in static water, in static wells, the detection of these neonicotinoids. And the detection at levels that ranged from at the level of detection, which is about 0.05 part per billion, upwards of about, I think here you can see there is one that's at 9 part per billion. These are low concentrations, there's no question. But we began to see the contamination in the static wells. And do understand that the Department of Ag has set up over the years static monitoring wells for groundwater height, but also for groundwater monitoring associated with spills or exceedances, right? So these are out of those groundwater monitoring wells. These are some of those levels. And you can see that some of these are squarely in the area of diversified vegetable production. Some of these are squarely outside of those areas as well. So, one of our research projects, and I did not put Anders' mugshot here, but Anders in the lab started to work on the question, can we change this? Can we consider different use patterns of these insecticides that limit or abate the flow through the water column? In fact, we thought that our ability to detect soluble insecticide moving out of this system might take as many as 1 or 2 years, just because of we didn't know anything about the solubility of these compounds, or not nothing, but we knew less about the solubility and mobility of these compounds. And what our interest was is that, are there ways we can deliver these tools and limit off-site movement? And so we were looking at things like foliar applications, seed treatment applications, the direct in-furrow application, which the majority of users will use or apply. They don't do it by hand, by the way. And then we were actually trying to consider the use of a polymer that could, whereby we could impregnate these compounds and hold them in place. And so that was the first half of this experiment. So we were applying them in this way, but then at the same time we were burying lysimeters below the soil profile to see if we can capture the soluble insecticides as they might be moving out of this system. And so that's the picture on the right, just to illustrate our experimental setup: a potato plant and then a lysimeter in which we hold it under a vacuum to draw free water in the soil into or through it. And then we would collect the water at regular intervals over time. I thought about not showing this figure, but it's one of those situations where, like, graduate students will always show you extraneous data. It's almost like look at what I did. Look at all of the stuff I did. Well, this is what I want to show. Digging these damn holes and burying them 3.5 feet in the ground underneath potato hills was no small task. A soil scientist would probably laugh at me at this point. But this was a randomized complete block design, again, imposing these different delivery systems and trying to see if we could limit or abate movement. So these are the actual in-plant concentrations of imidacloprid. This is one of the neonics here on the left. And thiomethoxam, another neonic on the right, applied in these different manners as the impregnated polymer, the in-furrow application, the seed treatment, a side dress application, or an untreated control. And thankfully, our untreated controls were nearly zero. That's an important point, nearly zero. But you can see that in 2 years of the study, 2010 and 2011, when you put a slug dose of insecticide in the ground and the plant picks it up, you initially get a high concentration, and then as the plant grows and there's less and less compound to pick up, it slowly trails off. So these are the in-plant concentrations above ground that we would see. I'd love to say that polymer just shone out as one that yielded the highest concentration throughout the season, but unfortunately it didn't. Again, remember the untreated control here is not zero. I don't want to say you could ignore the panel on the right, but the panels on the right are just showing you the cumulative inputs of water, whether it was irrigation water or rainwater, and then relative to vine kill. The inputs of water in both years were normal for the production season. The unfortunate part about this experiment is over here on the right. This is again thiomethoxam, and this is imidacloprid in 2 years, 2011 and 2012. And the dashed line is the time of vine killing. That is when we kill the potato crop, right? And these bars indicate the detection levels in mean concentrations in part per million of recovered pesticide. And as you can see, they stayed somewhat low, and about the time we vine killed, these came out of that system. And they came through those lysimeters within 115 to 120 days and arguably surpassed it. So, we did answer one question. We know these compounds can move more than 75 centimeters in a season. And so our question then was, well, this might be a problem. And in fact, this is now the emerging hypothesis, and that is that the majority of loss is later in the year, not earlier in the year that we had originally thought, and a lot of the product moves out of the system late. And so I'll go back to my comment about the untreated controls not being zero. Well, how could that be? I mean, the untreated controls not being zero would mean that, oh, well, okay, Russ, you've set up this experiment in a field soil where they've used these insecticides in the past, and what you're detecting is residual concentrations that are coming out of the system, and that could have been the case. So I asked Anders, I said, well, one other control that we should check is that this is in a commercial field where we have an operating pivot. I said, we better check the operating pivot and make sure that that's a zero. Unfortunately, it was not a zero, right? So the Department of Ag continued their pursuits after some of this work, and they have now looked at 87 static well sites where they've detected now 3 of these neonicotinoids at, again, 107 positive site year combinations, 45 clothianidin, 90 imidacloprid, and 138 Detex for thiamethoxam. Maybe there's a little bit of good news here. So the Wisconsin Department of Ag would set up a groundwater protection standard as part of law in the state, and enforcement based on the establishment of an enforcement standard, and that is again based on a daily consumption of about a liter of water per 10 kilograms of body mass. Think about that. That's drinking a lot of water in a given day. But this acceptable daily intake would lead us to an enforcement standard of 120 part per billion of thiamethoxam. We're nowhere near this, right? We're nowhere near these concentrations in the groundwater, right? And in fact, they would set up a preventative action limit, which is 20% of that enforcement standard. So the Department of Ag will not really react to these concentrations until we reach or exceed 24 part per billion. So from a human health standpoint, maybe we can say that we're okay now. This is a question. So you probably are aware of the geomorphology of central Wisconsin, right? I mean, the reason we grow veggies on sand is because when the glacier receded, it left this great inland lake, right? And you probably do understand that the Wisconsin Dells, right down here, were created when this lake broke through and drained, right? So veggies are grown on the east side of the lake shore, and cranberries are grown in the western shore, right, of the central sands. This is a distribution of the high-capacity wells in central Wisconsin associated with most of our vegetable production. And so because our untreated controls were not zeros, we decided to ask the question, so how extensive are these contaminants below ground in our operating pivots? And so that's what Ben, again in the goofy hat, has undertaken to work on. So he is actually, with consent, and this is important, with consent of the Wisconsin growers. I would hope to impress upon you that actually Wisconsin agricultural producers are probably some of the most forward-thinking individuals, I would say, nationally. They recognize that there is a potential issue here, and they are not trying to abate this work. And so our question is, how much spatial variability exists in these detects? And how much annual variation exists? So here we are looking at 3 test areas. The Anago Flats, which is where we grow seed potato. The Central Sands production area, which is where the majority of intensive vegetable production occurs. Sweet corn, peas, carrots, onions, potatoes. And then the lower Wisconsin River Valley also where some vegetable production occurs. Here again is a distribution of the high capacity wells, right? It's to me, just looking, it's amazing to look at this figure, I think. And but it is a testament to the extent to which agriculture is an important component in Wisconsin. And so here's really our test areas and where we have assayed these well clusters. So beginning in the fall of 2013, we assayed 48 operating pivots. In July of last year, 53 pivots. In September of last year, 26 pivots. And now in 2015, that number is up around 75 pivots. It's not 65 when Ben created this figure. And so this is just an illustration of some of those detects. Interestingly enough, we have about an 85% detection rate. So out of about 90 operating pivots, about 85% of those have detectable concentrations of a neonicotinoid insecticide moving through them during every irrigation cycle. We have higher concentrations in some of the northern production areas. We have a mean that's a little bit less in the central production areas and a mean that's even slightly less in the southern Wisconsin production areas. I would say the first principle of being a plumber applies here, and that is that poop runs downhill, is probably operative here. We probably do have a dilution issue here that is contributing to these lower overall detects as we go down into the drainage basin of the Wisconsin River. If we even look a little bit further, we can see that there is some interannual variation associated with season. We get a slightly higher concentration during the production season. In September, it's a little bit less again. But one thing important to note here is we have clusters of wells that we're looking at, and we can see a lot of variation among or between clusters of these wells. It's not homogeneous below ground, which is somewhat interesting. And then if we even look further, again with season superimposed and the individual well IDs, you can see there's a lot of well-to-well variation and to some extent some seasonal variation in those detects. And really the next step in this investigation is now to use what you maybe don't, or what you're seeing over here on the left panel is actually the USDA cropland data layer. It is Cropscape, which gives you 30-meter resolution on crop ID or parcel ID of 114 different land classifications that we will now begin the process of seeing if we can see relationships between the detects that we have and the cropping history that we have. This will be starting to get to a bit more of the punitive nature of this kind of investigation, because you might start to point at some crops more than others. And again, it is interesting that seasonally we see some variation. I mean, individual wells from one season to the next you can see one and in some cases 2 orders of magnitude change in these concentrations. So again, that might lead us to believe, well, either there's dilution that's washing the compound away, or there's variation in the magnitude of input of new concentrations of insecticide into the system, which we don't know at this time. So again, just to summarize Ben's work, these insecticides have been unfortunately detected at very low concentrations in a vast majority of wells. We do see some regionality with respect to those DTECs. Somewhat higher in the upper Wisconsin River watershed, somewhat lower in the southern region of the lower Wisconsin watershed. Again, we need to learn what sources of variation contribute to those range of DTECs. And again, our first approach will be to look at the crop IDs. And then temporally, we see a lot of variation just from one season to the next of an individual well. So we're getting close to the end here, but I want to tell you that these are data that are provided by the Wisconsin Department of Ag, Trade, and Consumer Protection. This is an illustration of their exceedance surveys that they've conducted from the mid-'80s until now. And I guess what I'd like you to understand is that the legacy of agriculture is right beneath our feet as far as the inputs that have gone into this system. The herbicides, the nitrates, the insecticides that we've talked about today, and to a lesser extent, fungicides. Do understand this is not an unbiased survey. This is the Department of Ag's survey, again, based on a spill or an exceedance. And they only have certain protocols for certain chemistries. So I'm going to say this is a fraction of what's actually down there. So to me, this is a bit of a challenge. So I said from a human health standpoint, we're in good shape. So those of you that had your hands up when I asked you about the word neonicotinoid, and then I asked you to keep your hands up if you heard of it in maybe a good light, all the hands went down. This class of insecticide is being implicated as far as issues with colony collapse disorder or bee disappearance. Unfortunately, the feeding bioassay studies that can result in bee disappearance, that phenotype results with feeding bioassays of 1 part per billion. So this is a challenge. This is a challenge. When the EPA reviews and evaluates new insecticides and new registrations, as they've always done, they've historically looked at the acute and to some extent the chronic effects if they have the tools in hand to do that. It's only now that we're realizing the potential chronic effects. The Deputy Administrator of EPA, Jim Jones, has said that this could be the legacy issue of his time as far as these issues with bees. And for sure the EPA is reacting because any time that insecticides are registered, they go through a reevaluation process on some schedule. The moment that the neonics were starting to be implicated in bee declines, they moved that schedule up. And they moved that up and they're under review now. So remember, we looked at this picture at the beginning of the presentation. I'm going to argue that vegetable production and specialty crop production in this country is at a time where it's getting ready to change. There's just not much water out here anymore. There's increasingly changes in precipitation patterns that are influencing the Pacific Northwest. The boom or bust meteorological patterns that South Texas faces is a challenge. Urbanization and salinity are unquestionably affecting production in Florida, and urbanization in the Delmarva is quite a challenge. Again, almost all of the canned vegetables in the grocery store come out of this area of the country and likely will continue. And this is an issue that we have, I'm going to say. This is a point in my presentation where if I'm talking to growers, I'm going to say, well, call me, consult me, and we'll figure out better options. I am going to say that solutions to these problems have existed for decades. With tools such as transgenic crops, cisgenic approaches, genome editing approaches now, would completely have revised that environmental profile, those level of contaminants that we would have seen associated with potato production. Remember, 1992 was when new leaf potato was registered in Wisconsin. 1994 was when McDonald's declared it Frankenfood, and it was stopped. So there are solutions out there. There are solutions that could have some of the single greatest impacts in changing sustainability, I think, in agricultural production. And still, we will not entertain their use. These are the growers in central Wisconsin. These are the growers that have to face the issue of what to use based on what's currently registered. And they are trying to do the right thing, right? So with that, I'll leave you, and if there's time for questions, I'll take them now.